Hello and welcome to this conversation with Dr. Bachner. It is Howard Bachner. I'm here with Francis Collins. Francis, welcome.
Nice to be with you, Howard. We can have quite a conversation here, I think, about what's happening with research and COVID-19.
We can. So for our listeners, Francis is director of the National Institutes of Health, probably the most influential, research-intensive biotechnology group in the world. It has a budget of $42 billion. Just remarkable science, lab-based, preclinical, clinical. Francis has been director since 2009. He came to the NIH in 1993. But Francis, before we get started, we're going to talk about the viewpoint that we released yesterday, and you've written with Paul Stoffels from Johnson & Johnson, entitled Accelerating COVID-19 Therapeutic Interventions and Vaccines, Active and Unprecedented Partnership for Unprecedented Times. But before we get into the viewpoint, Francis, you had a life before you came to the NIH. Could you tell people what your life was before you came to the NIH?
Oh, sure. It does seem like a while back, but I'm a physician. I went initially into physical chemistry was my calling, and then I got excited about life science and went to medical school at the University of North Carolina. I trained as an internist and then got my research training in molecular biology and human genetics at Yale, and then ended up at the University of Michigan on the faculty for 9 years in Ann Arbor, a place I loved. And my lab at that point was focused on trying to find the causes of genetic diseases for which we really didn't have any kind of functional clue about what the gene might be that was causing that disease when it was misspelled. So we chased after particularly ones that seemed common enough that you might have a chance of doing this with something called positional cloning. Before there was a Human Genome Project, before you could just go to the internet and read off those letters. And we co-discovered the gene for cystic fibrosis in 1989 with my good friend Lapchi Choi. His lab in Toronto and my lab in Ann Arbor basically fused together to try to do this. It's ironic because now when I think about it, what took us all of those years could be done by a pretty good graduate student with a thermal cycler and some DNA samples in about a week.
Yeah, pretty big change.
Big change. And we found the gene for neurofibromatosis, which for me as an internist who cares about genetics, about the most common disease that we see in adults that are dominantly inherited, and participated in a wonderful collaborative effort to find the Huntington's disease gene a couple of years after that. Yeah, that was my engagement. And yet when the Genome Project started to be talked about, I was a big fan because I knew we were never going to be able to extrapolate beyond these very difficult gene searching experiences if we didn't have a better map and if we didn't have the sequence to work with. And why not just do it once, make it public, and then empower everybody? And then that was pretty much the argument for the Genome Project. And it got started, and Jim Watson was the director, and then all of a sudden he wasn't. And then they asked me to come and serve in that role. I do recall that invitation as one of those things where I had very mixed emotions. What? Give up Ann Arbor? What? Become a federal employee? What? My mother always told me, son, you can be anything you want and I'll support you, but don't ever become a federal employee.
What were when you come to the NIH in 93, what were the early years like, Francis?
You know, it was pretty intense because I was charged not only with getting the Genome Project up and going, but starting an intramural program in genomics and genetics, because there really wasn't a lot of that going on at NIH at the time. So I had a lot of recruiting to do, a lot of building of programs. But for the Genome Project, I mean, we were just this baby in the crib at that point, trying to imagine growing up and actually succeeding and reading out all those 3 billion letters of DNA. It was a push. And it seemed at many times like we just were never going to get there. The technology was woefully short of what was going to be needed. So a lot of the effort had to be building new technologies for doing DNA sequencing and mostly focusing on simpler organisms where you could show progress, but you didn't have to tackle such a huge target. It was a good several years before I believed this was not going to be a disaster. And it really finally sort of, once it picked up, like with most large-scale projects, most of the real sequencing got done in the last 18 months, even though it was a 13-year project.
Yeah, I'm sure when you first arrived after the first few years, you were wondering if your mother was right. But we'll put that aside. So we'll talk about the Viewpoint, which is this private-public partnership, which was announced a few weeks ago. But Francis, what's the role of the FDA in a pandemic? Now, this pandemic is very different than Ebola or even when we've had very, very difficult flu seasons. But what's your vision of what the NIH role is? Because up front initially is the CDC, the FDA, less the NIH. So what do you think of as the principal role?
Well, we are the major supporter of biomedical research in the world, but that's the important term, research. We do not go out and do healthcare delivery. We don't do surveillance of what's happening across the country like the CDC would be doing. We don't do regulatory oversight of whether something is good enough to be made available to the public, whether it's a diagnostic test or a therapy. But we are partners in every way. We work extremely closely with CDC and FDA, and I have joint leadership meetings with their leadership. NIH's role in this pandemic, as it has been in others, but this time with particular urgency, is to try to figure out how can we accelerate the development of treatments and preventions like vaccines and better diagnostic tests that we're going to require new technology. All the things that you want researchers to do to really rise to the occasion and see what we could do to shorten what otherwise is going to be a painfully long process of death and suffering from people getting infected with this unexpected new coronavirus. So it very much all hands on deck. I mean, beginning at the very first moment in our vaccine research center, when it became clear this might be a threat to the world, and the viral RNA sequence was made public within a day. They had already designed the first vaccine, which was going to be RNA-based. And because of that speed with which that operated, that led to a world record by far, the first phase 1 trial getting started just 63 days later. That same trial, which just yesterday announced the phase 1 results, which look very promising indeed. And remember, this is May, and that was January, and we are now hearing about phase 1 results that are strong enough to say we're ready to go on to a phase 2 and 3, maybe combine those two together into a phase 2/3 trial. That's NIH at its best. That's NIH jumping on a research need and bringing to bear on that all the best technology and capable people that we could, but also working in a collaborative way with the private sector that has its skills. So this is NIH plus Moderna in this case. And I could give you many other examples, but that was what we felt we could do. Maybe not so visible at first, but now right in the middle of trying to be the solution to a terrible global pandemic.
So this will flow nicely into the Viewpoint, which is this partnership for unprecedented times. About 2/3, you know the data better than I, 2/3, 3/4 of your dollars go out, and a smaller percentage kept internally for NIH investigators.
Actually, it's about 83% that goes out.
83 goes out. I knew you know the percentage. So when you're faced with this new scientific question, how do you begin to think about using those dollars? Are you using those new dollars internally, as you did with the vaccine center? Or do you begin to direct external dollars outward? Or you do try to create this partnership? You have different options. I'm curious how you think through that.
Yeah, I try to think of all the above, because here's a place where if we have resources, let's not worry about exactly what the traditions were about which pot had which resources. Let's figure out how we can combine them to try to make things go faster. So what we're doing now in this partnership taps into that 83% that's going out to the extramural community that's going to be running clinical trials and has been and will be doing more. But it's also taking advantage of our own internal capabilities, particularly the Vaccine Research Center, but not just that. But again, let's be clear, as much as we think in this country, we're greatly fortunate and blessed to have all these resources for publicly funded research, and we are fortunate indeed. $42 billion is a lot of money.
It is.
The industry people are spending twice that every year. And we would be crazy at a time like this not to figure out how to put those things together. And that was really the motivation for assembling this group that ultimately became called ACTIV. Everything has to have an acronym, and it does too. Accelerating COVID-19 Therapeutic Interventions and Vaccines.
Now, I just wanted to let people know who some of the leadership. The co-chairs are you and Paul Stoffels, who's Chief Scientific Officer at J&J, and then members are Michael Dolston from Pfizer, Tony from the NIH, Gary Gibbons, NHLBI, Bill Peo from Roche, and then John Woodcock from the FDA. But then you have 4 subgroups, and I thought we'd walk through each of the 4 subgroups.
Right.
Preclinical, therapeutic, clinical, clinical trial capacity, and vaccines. Let's start with preclinical. Francis, what's your sense of what they will work on, or that group will work on?
So that group is particularly focused on trying to identify whether there are therapeutic agents that were developed for something else that have already been in human trials somewhere. So we know that they're not going to be really toxic, and they've already at least passed through that particular FDA approval process. And maybe, just maybe, somewhere in that pile of compounds is something that would have really strong effectiveness against SARS-CoV-2. So you're talking about repurposing a compound that was developed for something else. And how would you figure that out? Well, you'd need to screen those in a systematic way as possible against various assays, cell-based at first, to be sure that you're not missing something really important. And then for something that's really positive, you'd want to get them tested in an animal model. One of the things that group is doing right now, by the way, is to try to make sure that when it comes to non-human primates, where we need to test therapeutics and we need to test vaccines, and we have a limited inventory of animal model capabilities, let's not squander it on the wrong things. So we're trying to be sure that that priority setting is done properly. That's what that group is doing, trying to feed into the therapeutic pipeline things that you might not have guessed could be valuable, but would turn out to be just what you need based upon all the assays and all the science you can come up with.
Is that largely an NIH-focused initiative, or is that NIH with some of the private partners that have been created?
Well, the private partners are very engaged in that, and some of them are making available their particular compounds that have never yet been approved by FDA. Maybe they developed them for some other disease and they didn't look like they were going to work as far as efficacy, but they could be made available in this situation just in case they happen to have the right properties to be effective against this virus. So it's very much a wide open partnership.
Now, the second group is Therapeutics Clinical. We've heard quite a bit about different therapeutics, chloroquine, remdesivir, Toci, some of the other anti-inflammatories. How is the NIH and your private partners thinking about the therapeutics?
Well, they have a very busy time indeed. And this group, each one of these working groups is about 25 people, half and half from industry and from NIH and from academia, sort of combining those two together. And they working oftentimes at least once a day and gathering things in between meetings. They're meeting at night. They're meeting on the weekends. This is flat out, and the amount of dedication is amazing. Their first task was to say, okay, what are all the suggestions that have been made about therapeutic agents that might benefit COVID-19? Well, how many do you think there are? More than 600.
Yeah, right.
Okay, we probably can't put all of those through a clinical trial in the next month or two, so we better figure out which ones have the greatest promise and try to be sure we're prioritizing. So they started with 600. They got that down to 170 by sort of throwing a few aside that didn't have strong arguments. And then they narrowed that further to a little less than 40. And then ultimately, they were asked to come up with maybe 6 or 8 that would be the most compelling, that would have the strongest case for doing a clinical trial now. And they have worked very hard on that, and they put forward their draft proposal just about 10 days ago. And our executive committee will be debating that tomorrow morning or tomorrow afternoon when we meet to see whether they agree with those highest priority proposals, which is about 6 different therapeutic agents. And if they do agree, those will then need to go quickly into large-scale clinical trials. Don't want to wait. This is time to find out.
I think about 100 of those ideas have crossed my desk and submitted viewpoints. I've mentioned I regret not having kept a list because it would have been great to publish at the end. We haven't published many viewpoints about speculation about which drugs people should try.
You might have fun because we are actually compiling a list of the 10 worst ideas.
Yeah, right. That may be a more amusing list for people to read.
Amusing, but very stiff competition, I must say. It'll be hard to narrow it down to 10.
It may be. Clinical trial capacity. This comes up repeatedly, in part because of basket designs, adaptive designs. What's happened repeatedly in the past, you start a clinical trial and then the cases disappear. A huge problem. So how are you thinking through clinical trial capacity, Francis?
Well, again, we want to be sure if we need to do all these therapeutic trials, and pretty soon we'll talk about vaccine trials as well. Yeah. We want to know where the capacity is between publicly funded efforts through NIH, between such things as PCORNet, which is funded in a separate stream, and then all of the industry capability through CROs to do trials. We want to know what does that look like, that total inventory, and which of those trial networks could be ready to take on a new master protocol in less than 6 weeks. Because we not only want to know they have capacity, but that they're ready to start. That's what that group has been doing, is collecting that information. And it is impressive to see there are substantial numbers of such networks, many of them designed, of course, for other things, some of them for infectious disease, some of them for the lung disease, some of them just sort of general purpose networks. And when you put it all together, there is a pretty powerful engine there for doing these kinds of trials. And now we know where they are, and that's going to be helpful in doing the matchmaking between, okay, here's the compound you want to have tested. Where's the trial network that's best situated to take that on? A lot of it, though, as you say, you want to be sure that you're geo-mapping this onto where SARS-CoV-2 is currently circulating, right, where the cases are. Otherwise, you have a great trial and you can't enroll. So, and that's a moving target. So all that has to be sort of fitted together. Nobody's ever really tried to do this before for our entire nation's clinical trial capabilities. It's always been sort of piecework. And this time we're trying to do it all in a much more comprehensive way. And everybody's on board with that.
Francis, one of the things that I think is often confusing to the public is prevention trials and then treatment trials. Theoretically, the remdesivir announcement from the NIH was encouraging. It's a 5% mortality benefit, which is really important for that 5%, but it's not a prevention trial. Do you have a sense of the 6 or 8 compounds that you're going to talk about tomorrow? Are they balanced between prevention and treatment, or are they largely focused on treatment? Or do you
they're mostly on treatment. Actually, that group did have a substantive debate about where should their priorities be in setting their sights on what should go next into trials. And they still felt that the number one priority had to be people who are sick. And so these are primarily focused actually on people who are sick enough to be admitted to the hospital. Recognizing that we really also need help for those that are ill but not ill enough to require hospitalization, and we need things for prevention. What's coming, though, pretty soon, Howard, in that regard, in fact, this is now the big focus of that same working group, is what are we going to do about monoclonal antibodies? Right. Many of us, I think, looking at the landscape would say, if there's a hope for something with a big effect size, remdesivir was like, okay, but let's have something that really gives you a big benefit. I think there's evidence based on other circumstances, but nothing you could be sure of, is monoclonals derived from people who've survived COVID-19 ought to be our best hope here. Because we know the immune system is pretty good with this particular virus. People get over it, and you can't find the virus anymore once they're over it. So that means antibodies and maybe some cell-mediated immunity are pretty darn effective. If you could deliver that passively, you'd probably have something pretty good. Now, that would, of course, be applicable both to outpatients and inpatients. So I will tell you, we're already in the process of designing a master protocol for monoclonals for outpatients and a separate one for inpatients. And there are more than a dozen companies now that are on the process of developing monoclonals for clinical application at various timetables, but some of them are getting pretty close to being ready. And again, we would want, because we're all in this together, to do that in a fashion where you have some harmonization of those protocols, where you can do comparisons across trials, where you have standard endpoints, where you have an understanding about what lab measures you're going to use to decide whether it worked or not. Everybody's pretty committed to that, even though not all of these monoclonals will come along at the same time. That's going to be a big test of whether therapeutics can really rise to the occasion here. And I'm guardedly optimistic we're going to see something encouraging.
Yeah, we've actually solicited a viewpoint from a number of people from the NIH on monoclonals, in part because I think there's been some coalescence in the last couple weeks that in the short run, vaccines are still a longer play, but that in the short run, let's say the fall, when you really want people to go back to school and college, we talked about this, monoclonals, you could deliver a large number and you could probably get them to the market sooner if they're safe and effective. And,
yes.
and so I think we solicited a viewpoint because we feel like our role is partly to educate people about what people are currently thinking.
Yes.
The fourth bucket is vaccines.
Of course.
In the newspaper every day, particularly this morning, controversial about the Healthy People Challenge. But let's just start with vaccines in general. Then I'll have a few more specific questions, Francis.
Sure. Well, obviously, everybody wants to see this go forward as swiftly as possible, but also to be done in a fashion where you can be confident that if something does come through the trials, it's not only effective, but it's also safe. So we don't want to cut corners in this space and end up with something that hurts people. And because you're talking about giving vaccines to healthy people, your standard has to be really high about safety. But how do you do that and not end up taking 2 more years to get to it, which would be a more standard approach, or maybe 3 or 4 years? Here again, I think everybody has to figure out ways to accelerate that process without sacrificing the rigor. One of the ways, of course, is to start early. And I mentioned already what was done in terms of this NIH Vaccine Research Center Moderna collaboration getting started and into phase 1 trials in just 2 months. And there are others that are doing that as well. You may have read about the Oxford chimp adenovirus vector vaccine, which also looks pretty good. And there are others not far behind. In fact, I would say there are at least 4 or 5 of these vaccine platforms that are looking as if they are going to be ready for a large-scale trial in the fairly near future, let's say the next 2 or 3 months. And you would want them to have that chance to see how that works. Ideally, again, we want to do this in a fashion where you can learn from each trial things that will benefit an understanding of the whole process. So we don't necessarily want these to be on completely separate pathways, but doing what we might call a harmonized master protocol, again, where you're sharing information about the endpoints and the surrogate measures and so on. And that has pretty much been agreed to. I think we will see the initiation of these trials as early as July. The FDA, I think, being very much on board here with trying to do this in an accelerated pace, sees this as an opportunity to go from phase 1 to something you might consider sort of a combination, phase 2, 3.
Yeah.
So basically, you're watching very carefully at the beginning of a phase 3, and your DSMB is looking to see is there any signal there of trouble, effectively acting as if it was sort of a phase 2. But you're not going to stop. You're just going to keep going if all things are going well. So potentially then, if we can mount that, and if we can find, let's say, 10,000 volunteers for each vaccine platform, that's a lot of people. And of course, you have to find them in a place where the virus is actively circulating. You won't know if your vaccine worked unless it protected people. And you won't know that if the virus is not around them. So this means, again, doing this sort of geomapping of where your capacity is for vaccine trials and where the disease is. I think we can make that work, but it's going to be an enormous challenge logistically. The other thing you got to do is not wait to begin manufacturing. Because if that were your model, you might get to the end of your trial and go, hooray, we're going to have a press conference, our vaccine worked. And everybody goes, okay, now where is it? Well, sorry, we got 6 months to scale up the production. Just wait. That's not the right answer. Fortunately, the U.S. is in this situation, particularly of this organization called BARDA within Health and Human Services, to put money into scale-up manufacturing of vaccines even before you know if they are going to work. And I'm sure some of that will be wasted because some of them won't work, but hopefully some of them will. And then you'll be ready, maybe as early as the fall, with millions of doses of a vaccine that has at least received emergency use authorization from FDA. That's the hope. I don't want to be too over the top in confidence about this. This is white knuckles all the way. There's so many things that might go wrong here. But I can certainly tell you that everybody from the public sector to the private sector to the government is doing what they can to try to make that happen with no letting your foot off the accelerator, not for a minute.
Francis, do you worry about the ethics of challenging healthy people with live virus when there's not a cure? I mean, you know, there's been many different opinion pieces written about this. You and Tony and others are at the center of this discussion. I'm just wondering how you think about that.
Yeah. Well, I understand the appeal of it, because it certainly could be the case that such a challenge trial could be done in the fairly short order, at least hypothetically, and then FDA might be willing to say that's good enough. If you vaccinated somebody and then you intentionally infected them and they didn't develop disease, okay, that looks like success. But suppose it isn't so successful, and suppose some of your healthy volunteers who were healthy aren't healthy, and suppose you even have mortality. Yeah. It's very hard to see how that's justifiable for me when you have a disease where you don't have a rescue therapy if somebody gets sick anyway. If our monoclonal antibodies look really good in 2 or 3 months in terms of their ability to save lives of people who are really sick, then this could change the ethical circumstance. The other thing, Howard, one needs to say is that even if you thought it was ethically acceptable, don't imagine that this kind of human challenge trial can happen all that quickly. You've got to make a stock of GMP material. You've got to set up a circumstance where you can intentionally infect people, which means you're going to have them quarantined with their staff for weeks at a time. You don't want to set off a new episode of infection in the community and do all the measures. Most people would say we're at least a year away from that, even if we decided right now to start that effort. A year from now, we should have done it the right way with an efficacy trial in a community-based approach, and that would be much more convincing anyway. Last thing I'd say about the challenge trials is, who do you challenge? You challenge young, healthy people. Right. But okay, so if it works for them, does it really tell you that it's going to work for the elderly or for the people with chronic diseases, who are the people you most want to be able to assist? Or are you going to fool yourself into thinking you've got a really good vaccine and it doesn't make that much difference in the death rate? That would be horrible.
Francis, in these private-public partnerships, I know there's the National Institute of Health Foundation. You know, the issues of financial conflict of interest comes up, and I'm wondering, you know, you lead the most important research institute in the world with an enormous amount of resources, but as you said, the private sector in the U.S. contributes $75 or $80 billion, particularly to clinical research. Right. How do you think about the financial conflicts of interest?
We have to pay close attention to them because that can compromise everything we're doing in terms of not really having the kind of objectivity that it should, and certainly can do great harm to public confidence. I guess I've learned over my 11 years as NIH director that one needs to look closely at that, but you should not let it discourage you from partnerships that can be done in full openness of what's going on. Again, transparency is the best way, I think, to address the potential for conflict of interest, because then it's wide open there and people can see whether they think it's at work or not. This active partnership we've been talking about has 18 pharmaceutical companies involved, as well as numerous NIH institutes, FDA intimately involved in that, CDC, BARDA, the Foundation for NIH as a nonprofit serving as a program manager. It is wide open. There's nothing we're doing that I wouldn't want anybody else to know about. And it's certainly not leading to a place where there's any intellectual property that's being generated. But it is making it possible to go faster, which is what I think the public expects of us, than if we were all off in our own corners afraid to talk to each other. I've done this before with something called the Accelerating Medicines Partnership, focusing on Alzheimer's disease and diabetes and rheumatoid arthritis and Parkinson's disease and cancer. And those projects have been remarkably successful in advancing the field, but have made all their data immediately accessible to everybody. That just seems like the right thing to do when we're waiting for progress to happen. Why don't we bring everybody together and make it happen a little faster?
So just a couple other questions. So Francis, when you were at Michigan and you and your colleagues isolate the gene that identified cystic fibrosis. 25 years later, did you think there'd be a treatment?
Well, there was a great hope on that announcement in September of 1989 that it wouldn't take long, because we now finally had the gene therapy was looked at as like maybe that was going to be a quick one, if only we knew. And especially because it was a disease of the lungs and the airway, maybe you could just put the normal copy of the gene into a virus, inactivate the virus, have somebody take it as an aerosol, and all would be well. And much work was done by a few of us to try to see if that would work, and it was a total frustration. We underestimated the immune system's ability to recognize something that wasn't normally supposed to be there and to wipe it out. And so for the next almost decade, progress for therapeutics really, really was stalled. And then it was really the effort to try to change the paradigm from gene therapy to drug therapy that finally now has paid off. And much credit for the Cystic Fibrosis Foundation for the way in which they took a big risk on that, and to the private sector for Aurora, which then later became Vertex. And now here we are, Howard, in this amazing moment where 90% of people with cystic fibrosis have a triple drug therapy that is phenomenal in terms of what it can do in terms of turning around lung disease and preventing further progression. I get emails almost every week from somebody with CF who has had this experience of getting on these therapies. Some of these people who were pretty much getting close to time for a lung transplant and who are now back at work. It's amazing to see. When it works, what can really happen? That's so inspiring.
What was it like to grow up in the beautiful Shenandoah Valley and be homeschooled, Francis? You were homeschooled until 5th or 6th grade. What was it like?
Oh, now we're really going back, Howard.
We are.
It was wonderful. My parents were kind of hippies, except the hippies hadn't really arrived yet. Living on this farm in the Shenandoah Valley with no indoor plumbing, starting an outdoor theater in the grove of trees up above our house, which is now, by the way, in its 64th consecutive season, or it would be if they hadn't shut down for COVID-19. It was all about music and the arts and interesting people and literature, and science not so much. That was something I got interested in on my own. And my mother taught myself and my 3 older brothers at home because she was convinced she was a better teacher than what we were likely to encounter in the local county schools. And she was absolutely right. And so I got inspired by her about the joy of learning things. That was just such an adventure. It never occurred to me that people might find that to be hard work or boring. It was like every day was something new to discover. Finally, when we moved in town to live with my grandmother because she'd had a stroke and needed somebody in the house, my parents decided, okay, you're the youngest. We'll let you go to public school and see how you do. And fortunately, that was good, too, because public schools were quite good at that point, and I met science in a high school chemistry class that changed me in a very significant way in terms of what I thought I wanted to do with my life.
Well, you've done pretty well, Francis. There are quite a few questions, so let me go through a few questions and then I'll wrap it up. Monoclonal antibodies, optimistic. When do you think they could be available?
Well, there are at least 2 companies, Regeneron and Lilly, who say they're pretty close to being ready to mount into clinical trials. And obviously, then you have to see how long does it take to enroll patients and what does it take to show an effect size. I think we all believe that if this is going to work, the effect size ought to be substantial. So maybe the trials don't have to be that big. So I'll be optimistic and say maybe by August, we'll start to see results.
Antigenic shift in the virus has been on the front page of newspapers. It often happens, quite a few articles have passed my desk. Most people have said not dramatic, not substantial enough. I'm just wondering if you can comment on that, Francis, because it is concerning when the public reads, oh, the virus is changing. So could you just comment on that?
Well, yeah, first of all, if it weren't changing, that would really be news because this is an RNA virus. RNA viruses change with mutation over the course of time, especially if they have lots of ways to get passed around amongst lots of people. So the total population of viruses gets to be huge. This one seems to be traveling, as far as its mutational rate, about typically for an RNA virus, not extremely mutatable, but not extremely stable either. So far, what I have seen has not caused me pause in terms of the rate of mutations falling into vulnerable places like the spike protein that might have a big effect either on diagnostics or on therapeutics or vaccines. But you got to watch that. And it's a good thing we now have the capability to do that sequencing of lots and lots of isolates. So we will know. I mean, one of the big parts of this is how long will immunity last for people who've gotten over infection? And a correlate of that is how long will vaccines work? If we generate immunity to today's SARS-CoV-2, what will that look like 2 years from now or 5 years from now? Will that require a whole new vaccine strategy? This isn't as bad as influenza. Influenza clearly moves more rapidly than this coronavirus does. But it's certainly possible that this is not going to be something that will be stable over long periods of time. But in the short run, I would say we don't have any major reasons to be concerned that this is changing so fast that our strategies from yesterday won't work tomorrow.
In 2017, you wrote a piece for us for the Arts and Medicine section entitled Sound Health, an NIH Kennedy Center Initiative to Explore Music and the Mind. Frances S. Collins and Renee Fleming. I think most people would know who she is. You have two professed loves in your life, music and motorcycles. Why don't we start with music, and then you can comment on motorcycles.
Well, part of growing up in that Shenandoah Valley farmhouse was an exposure to music. My dad played a really good violin, and he could also play a mean hoedown fiddle if he needed to. And all sorts of musicians were popping up all the time to stay with us for days on end when they ran out of money. So music was part of what I grew up with. I play guitar, I play piano. I find this is a way to unwind and relax after a particularly intense experience. And I'm all about intense experiences right now. So I'm playing my piano quite often in the course of one of those very long days and nights. It gives me this sense of sort of being lifted out of the intense details of what's happening in a scientific life into some different plane. And to be able to do this with other people who I greatly admire, like Renee Fleming, has been an enormous gift. Unfortunately, so many of us who love music have been prevented from gathering together. I'm in a rock and roll band. We've lost 3 gigs already that we were going to be doing during this time, and they're gone forever. We haven't even been able to get together to rehearse. And I miss that a lot. But I know the day will come where we'll be able to bring this all back together. Hey, Howard, I will send you a little video I made for the NIH staff where a bit of a rewrite of John Lennon's song Imagine got put together. And I did a little solo video with my wife as the videographer, which has a little bit of a tongue-in-cheek about exactly what it is that we're trying to imagine. Now, I'll just send it to you. You'll
send
get
it
a bang
along.
out of it.
That'd be great. And motorcycles.
Yes, another indulgence. I started riding in college. I gave it up for a while when my kids were little because it did seem irresponsible, and it probably was. And then they got old enough, I figured, okay, I'm going back. And I'm on a Harley Road King Classic, and it is also a wonderful way to unwind when you just want to crank that accelerator down a country road and kind of let the rest fade away for a few minutes while the wind blows and your engine roars and you take a really nice corner in just the right way. Hard to beat that.
This is Howard Bachner, Editor-in-Chief of JAMA, and it's been a wonderful privilege to talk with Frances Collins, who's Director of the National Institute of Health. It's been a wide-ranging conversation about the NIH, its response to the pandemic, and we've been discussing France's piece that he's written with Paul Stoffels from Johnson & Johnson, Accelerating COVID-19 Therapeutic Interventions and Vaccines, Active and Unprecedented Partnership for Unprecedented Times. Frances, stay healthy. Thank you for everything you do for the American and international populations. Thanks, Frances.
Thanks. And thank you, Howard, for everything you do to get the word out about all the things that we're learning and need to learn more. Appreciate your leadership as a remarkable editor.
Okay. Bye-bye. Be well.
Okay. Bye.